Hello students, in this module I'll be talking about the second part of the TS of the anther, where I'll be discussing the 4 layers of the anther wall. So look, listen and learn. What you see here is basically just one microsporangium which is being enlarged. So I'm going to still call it as the TS of the anther. Is that okay? Now, what was the outermost layer? You got it. It is called as the epidermis. Yes? So the epidermis basically is the outermost layer, right? Next, you must know that it basically is made up of flattened cells, right? And finally, the main function of epidermis is going to be protection. Is that okay? Moving on. Same picture again. This time, I'll be talking about the second layer, okay? The second layer is called as endothecium. Is that okay? Now the endothecium basically lies internal to the epidermis. Next, you should also know that the endothecium is a common layer for all 4 pollen sacs or for all 4 microsporangia. Is that okay? And you should also remember that the endothecium basically is also made up of a single layer of cells. Yes? Now remember that the cells of the endothecium basically shows fibrous thickenings of a sugar substance called as callose. The name says it, callose, it has to be a sugary substance. Is that okay? Why it is important to understand about callose? Because remember this picture of the TS of the anther which was dehisced? Is that okay? Now remember that area which you see from where dehiscence will take place, the cells of the endothecium are very, very thin-walled. Is that okay? Yes? So that particular area exactly marked is the shallow groove that you see between the 2 microsporangia. And the cells here basically have a thin wall, and this basically represents the line of dehiscence. Is that okay? And please remember that the main function of endothecium is because of the callose present. So the callose is hygroscopic in nature, which means it will absorb moisture from the environment, right? Hence, the endothecium will help in the dehiscence of the anther at maturity. Because when the callose absorbs the moisture, it will swell up, it will burst open, the anther will dehisce, and the pollen grains will be released. So the endothecium basically participates in dehiscence of the anther thanks to the presence of callose. Is that okay? Moving on. Let us look at this next layer. This next layer is basically called as middle layers. Now the middle layers is internal to the endothecium. Is that okay? The middle layers can have anywhere between 1 to 3 layers of parenchymatous cells. Is that okay? And also remember that the middle layers surround each pollen sac or each microsporangium individually. Is that okay? Now, look at this picture. I've just shown you one half of the TS of a dehisced anther. What I'm trying to point out here is that area right there, right? Between the 2 pollen sacs. They say that the middle layer cells basically degenerate at maturity, and that is how the 2 pollen sacs merge and become one, and then your pollen grains are ready to dehisce, right? From that area of dehiscence thanks to callose in the endothecium. Is that okay? Fine. Now, there you go. What I've shown you here basically is how the 2 pollen sacs are merged into one. Is that okay? Moving on. Finally, you have the 4th or the innermost layer, which is called as the tapetum or the tapetum. Is that okay? Remember, it is the innermost layer. Yes. Then what you see here in the center is where the pollen grains or microspores will be produced. So that area is simply called as the sporogenous tissue. The tapetum basically surrounds the sporogenous tissue, right? And what is so special about the cells of the tapetum? You must know that the cells of the tapetum basically are larger size cells. Is that okay? These are cells which contain dense cytoplasm. These are cells which contain 1 or more diploid nuclei, or they may simply contain a single polyploid nucleus. Is that okay? These are all the points you have to remember about the cells of the tapetum or the tapetum, right? And finally, you should know that one basic function of the tapetum is nutrition. So it provides nutrition to the sporogenous tissue and the developing microspores. And the most important function of the tapetum or the tapetum basically is formation of sporopollenin. Now, sporopollenin basically is a component of the pollen exine. Is that okay? So what I've discussed here are the 4 layers of the anther wall, namely epidermis, endothecium, middle layers, and tapetum or tapetum. Right? So in this module, I've discussed the second part of the TS of the anther. In the next module, I'll be talking about the microsporangium, and I'll also basically be talking about the structure of the pollen grain. Till then, thank you and take care.